Ever wondered how scientists decode the molecular language of cells? Or why AI models might flip their answers based on how they're built? Today, we're unpacking a groundbreaking study that uses deep learning and SHAP, Shapley Additive Explanations, to reveal how biological data is interpreted, and why those interpretations can be surprisingly unstable. Let's start with a big picture. Multi-omics is like peering through a kaleidoscope. Proteomics proteins, metabolomics small molecules, lipidomics fats, and more. Each layer offers a unique view of a cell's activity, but integrating them is like solving a puzzle with missing pieces. Deep learning models, which mimic neural networks, have become powerful tools for this task. But here's the catch: they're often black boxes. Even if they predict accurately, it's hard to see why until you bring in SHAP. SHAP is the detective of AI. It assigns scores to features, like proteins or metabolites, showing how much each contributes to a model's prediction. Think of it as a chef tasting a dish and identifying which ingredients made it exceptional. But here's the twist: this study found SHAP's results aren't always consistent. When researchers tested different deep learning architectures, think of them as recipes for building models, they discovered the same molecule could be flagged as critical in one setup, but irrelevant in another. Even tiny changes, like random weight initializations during training, could shift rankings. It's like if a recipe's success hinged on whether you stirred clockwise or counterclockwise. To dig deeper, the team used real-world data from influenza studies. They tweaked model structures, layer counts, and training seeds, then tracked how SHAP scores changed. The results were sobering. Feature importance varied wildly. A biomarker deemed vital in one run might vanish in another. This means relying solely on SHAP could lead to false conclusions, like assuming salt is the secret ingredient when it's just one of many. But the study didn't stop there. The researchers proposed a solution, testing features across multiple model versions to separate true biological signals from random noise. It's like baking a cake repeatedly to ensure the recipe works, not just a lucky batch. Their experiments showed that while some model adjustments stabilized SHAP scores, they often came at the cost of predictive accuracy. It's a trade-off between fairness in feature rankings and the model's overall performance. So what does this mean for biology and medicine? If AI is used to find disease biomarkers, we need caution. A feature labeled important today might not be tomorrow, depending on the model. This work underscores the need for transparency in AI research, especially when stakes are high, like human health. Here's the kicker: even with the same data, different training runs could flip rankings for the same features. It's like if every time you made a recipe, the importance of salt changed, sometimes key, sometimes negligible. This sensitivity highlights how fragile these models can be. But there's hope. The study suggests balancing model complexity with data type, and combining AI insights with biological knowledge to filter noise. The takeaway: deep learning is powerful, but it's not perfect. Understanding how noise and compression affect feature importance is crucial for trustworthy results. Next time you hear about AI solving complex problems, remember, even the smartest models need rigorous testing. Thanks for watching. Let's keep the conversation going. Have you ever wondered how AI decides what's important? Drop your thoughts below.